All right, y'all. So it looks like it's going to be an intimate group of 1, 2, 3, 4. Looks like there's 5 of us. I think I only am familiar with one person on. So I'm just going to get started now. I'm going to just freestyle it. It's going to be a shorter session than we usually have. So I just ask if before we get started, if you guys mind just introducing yourself. Quick facts: name, first name. Don't even need to give us your full name. Where you're located. I'm in Brooklyn. And one more thing that you want to add, it could be where you went to undergrad, your ideal medical school that you want to go to, or if you want, for an exercise that we're going to do later on today, you can just share what kind of doctor you want to be. So I'm going to start, and then I'm just going to unmute one person at a time and just call on you. So my name is Kamanthi. I'm a second-year medical student at SUNY Downstate in Brooklyn, as I mentioned. I am from Raleigh, North Carolina. I went to Duke University. I was in engineering college. I've done a post-bac. I'm a non-traditional career changer. I'm turning 29 in a few weeks. And I took a very unique path to med school. Studied for the MCAT in the summer of 2018. Originally planned on taking the exam in May 2018 because that aligned with the completion of my first year of my post-bac program, where we were mainly taking, chemistry, physics, biochemistry, those kind of courses. So in planning my transition as a non-traditional, I'd kind of planned to take the exam in May. I wasn't ready. I ended up moving my exam back to the last date in June 28th. And I tackled MCAT studying by trying to break it down and understand what it was about. I kind of leaned on my natural quantitative engineer coding skills, if you will, to sort of crack the code of what I need to do to kind of go from the 501, 495 range I really was at to the 515 score that I needed in order to really prove to the med schools that I deserved to make it into an interview. So that's my introduction. I kindly ask that we just kind of round robin. So Angie, I'm going to go ahead and ask you to unmute. And if you don't mind, just introducing yourself, nothing as long as I did, but the basics.
Okay, so I'm Angie. I recently moved to Chapel Hill, North Carolina. And
wow, Chapel Hill, shout out to you.
yeah.
I don't know if we can be friends or not, because I went to school and I'm a big Blue Devil fan. So I don't know if are you from North Carolina family, or is it just like a move for a job or something?
So I'm from Clearwater, Florida. I grew up and lived there my whole life, and I moved here a month ago because my boyfriend, he's going to UNC for pharmacy school. So I'm just here with him during my application year until I figure out where I'm going for medical school.
Okay, wonderful, wonderful. Thank you very much. And what's one thing that you want to get out of today's session?
I would like to build on my critical reasoning skills.
Okay, great, awesome, thank you. And then let me go ahead and ask Christy, and Angie, just unmute yourself back after you're done. Christy, do you mind just unmuting and introducing yourself?
Hi, I'm Christy. I'm from San Jose, California.
Mm-hmm. And tell us a little bit about yourself.
Yeah, so I graduated and I'm currently taking a gap year to study for the MCAT, but I've been kind of like struggling with approaching like experimental passages and science passages.
Mm-hmm.
I'm hoping to learn more about like strategies for the session.
So let's call it critical reasoning as well.
Critical reasoning.
And I'm purposely saying that because that's going to tie into a lot of what you get out of today. So thank you very much. And Victoria, do you want to unmute and just kind of introduce yourself?
Yeah, I'm Victoria. I'm basically near San Francisco, and I go to UC Santa Barbara. I'm going into my last year, but I plan on taking a gap year before med school. What kind of doctor? Maybe pediatric endocrinologist, but I'm not sure.
Ooh, Victoria, I like that. Pediatric?
Yeah, I like
endocrinologist. All right, so you said pediatric, so we're going to just remember, I'm going to write a note, and then I'm just going to remember that for an exercise we're going to do during the session today, okay?
Sounds good.
All right, cool. So who's next up? Jaleesa, you want to introduce yourself real quickly?
Hi, I'm Jaleesa. I'm from Tampa, and I am working with the homeless for the year, and I want to do family medicine.
All right, awesome. Family med, great. And last but certainly not least, I don't want to pronounce your name incorrectly. If I do, please let us know
okay.
the right way.
I'll just pronounce it for you.
Okay, awesome, awesome.
It's Irena.
Say that one more time.
Irena, like your ear.
Oh, Irena.
Like your ear, Irena.
Irena.
Yeah. So I'm Irena. I am from southeastern Ohio. I was born and raised here. I'm 29. I attended the Ohio University. I graduated in 2013 with a BA in human biology. I have been working as an EMT for 4 years, and what I hope to get out of this session, I've taken 2 practice exams this week, and I'm running out of time because I don't understand what I'm reading. So definitely comprehension and critical reasoning skills.
Okay, awesome, thanks. Is there anyone else on the line that I forgot? Okay, great, awesome. So typically I go through a long spiel to basically explain a very simple question, right? So what do you think the MCAT is, right? So anybody on the line just kind of want to just hop on and just kind of give us an idea of what they think the MCAT's really about. Like honestly, no wrong answer. Well, there is a wrong answer, but I assume most of y'all are going to say the wrong answer. So feel free to say it. What's the exam actually testing you? Because like it's a really important thing to know if you're going to prepare for anything, right?
It's critical thinking.
But there's an even bigger part of it, right? One thing I've realized now that I'm like going into my second year and I'm reflecting on 1 year of med school, 1 year to go, step 1 ahead, and a year and some change of MCAT tutoring is what the MCAT was really trying to see, if you were able to do, is are you prepared to begin to learn how to reason clinically? It's really like a pseudo exam for your clinical reasoning. It's your first clinical reasoning exam, okay? Is anybody on the call is anyone confused when I say the term clinical reasoning, or that the exam is really trying to test clinical reasoning? And if you are, it's fine, because we're actually going to dig in and practice some clinical reasoning, not critical reasoning. We're actually going to practice clinical reasoning today. So if anyone's confused, just feel free. Like this is like small, intimate group. Let's make this conversational today. Okay, quiet group, no problem. So I'll just call on somebody. How about you, Angie? Angie, let's chat. All right.
Hi, sorry.
When I say clinical reasoning, what does that mean to you?
So I feel like that has to do with picking out the most important information from the passage, because you're thrown a lot of words, and I know they're not expecting us to understand everything. So maybe it has to do with picking out the important pieces to come to an answer.
Wonderful. And how does a clinician, give me an example how a clinician, right? Give me an example how the future you is going to perform clinical reasoning when you're sitting in a white coat at, where would I put my notes? When you're sitting in a white coat in, let's say you decide to go to UNC, right? You're sitting in a white coat in a year. Give me an example of you practicing, or even longer than a year, an example of you practicing clinical reasoning.
So would that be where like the patient gives me like their whole story when they come in, and then I just ask follow-up questions? Basically?
You ask follow-up questions, right? So you basically went into that interview with that patient with a list of questions to ask them, right?
Right.
What are the questions that you go into a practice passage looking to ask or get answered? Do you have any list?
Wait, I didn't understand the question, sorry.
So when we're talking about clinical reasoning, right? And you said clinical reasoning is like when I go to a doctor, or I'm a doctor and I'm asking a person a question and trying to figure out what they are saying, right?
Right.
And you said, of course, you have to ask them probing questions, right? So I'd assume if you're, a medical school student, you study, you're a hard worker, so you know that there's specific questions you got to learn for your physical exam or your medical interview, and you study that list of questions, right? Why? Because you got to go into the room and do exactly what you said. You have to perform clinical reasoning, right? So what I'm getting at is a very simple rhetorical statement, right? When you go into an MCAT passage, and this is for everyone listening, do you have a plan already before you hit the MCAT passage? If not, right, ask yourself how you would feel if your doctor walked into the room with no plan before they we're about to perform some clinical reasoning on you. Does that make any sense, guys?
Yes.
Right? Makes sense, right? It makes sense. Like you want to be prepared, right? So that's all I'm going to do in terms of setting the stage for kind of explaining the background of what we're going to do. So we're going to go ahead and start this practice passage right now, and we're going to do it collectively together, okay? So in order for us to do it, I want, we're just going to go round robin. Angie, thank you. So Irena, did I say it right? I got really bold.
Yeah.
Wonderful. So why don't you step up and why don't you tell us what you think this passage that I got up here is about? Give me like what's your first initial take of what the whole passage is about.
So this passage 25.
Mm-hmm.
I didn't read this one. Should I have?
Well, give it a look. Just take a look at it, 2 seconds, and tell me what stands out with you. And in 5 seconds, tell me what you think it's probably about. And while you're doing that, why don't you tell me what you would do if this was a real exam and you had a few minutes, few seconds maybe, to figure out what an actual passage was about. Maybe it was a tough chem phys section, and it's towards the end of the exam, right? So I want to also get you into the habit of articulating your plan.
Okay.
Right. So I asked you a simple question. What's the main topic? Tell me what your plan is, and then answer what the topic is about.
Give me like 30 seconds to read the first paragraph.
No problem. And I'm going to go ahead and give you a little hint. I'm purposely trying to stress you to kind of drive a point home. So stay calm and keep going.
Okay.
And all of you guys and the rest, like, feel free to like answer and respond and collaborate either in the chat or like regularly, like by talking. It actually makes the session a lot better. So if you've already figured it out, just put it in the chat. It's going to help me know if you guys are all engaged because it's going to be important. Mm-hmm. Okay, Victoria, I see an answer from Victoria. Angie's got something. Jaleesa, what do you think? Cell structure, okay.
Okay, so I just read the first paragraph here.
Okay.
The first thing that I would highlight is eukaryotic cells.
Why?
Because it's important for me to differentiate what it's talking about. So if I were to confuse this with a prokaryotic cell, which is a mistake I've made in other passages, so that would be something that I would highlight.
Mm-hmm.
And it goes into talk about, you know, the microtubules and microfilaments and the function of them. Something else that I found maybe of importance when it talks about alpha beta heterodimers. So maybe that's something else I would highlight.
Mm-hmm. Okay, no problem. So I wanted to start the exercise here so we can actually take a step back, right? Because I told you need a plan before you start, but I never actually shared my plan, right? We're going to start the plan right here. We're going to start the plan by simply understanding the type of science passages they are, right? So I like to say there's really 2 main types. There's 3. Obviously, there's a persuasive, but I don't really do much and teach much about it because it's not really that big of a deal. If you understand the first 2, you'll be good. The informational passage is really a chem phys passage, the dominant passage in chem phys, right? So now that I said chem phys, I want you to just take a second and let some chem phys topics rattle in your head. And let's just kind of start getting some flying in the group chat, right? So I'm going to start one off, like magnetism is one, right? So start like literally like write down in the group chat right now any chem phys topic that comes to mind content-wise. Fluids, optics, electricity, any more? Circuits, kinematics, right? We got a couple. Forces, cool. So what I'm trying to get you guys to start to do is kind of start to think about just general scientific information, right? Really, I'm actually trying to get you to think about really straightforward scientific information that you might already know. That's a key thing to understand with a lot of chem phys passages, right? You might already know the general, like all the science that's associated with the passage, right? Let's say, for example, electronegativity, right? You might know something about electronegativity, right? So somebody say something about electronegativity on the line, like get on 3 quick facts.
Periodic trend, it goes up the periodic table and goes to the right. That gives you increasing electronegativity.
Okay, trend, that's one thing. All right, somebody else want to come in and say one more thing? I saw fluorine. I'll give you fluorine because I really do think fluorine in my mind is a separate thing for electronegativity, but really it's a trend thing. But let's get one more thing. I'm going to add one. I'm going to add ionization, right? And I'm going to add ionization. I'm going to keep it open. Does anybody want to know or say why I added ionization as an extra topic related to electronegativity? So quick moment to capture a content level piece of information. So how did these two ideas square up? Ionization and electronegativity. Anybody? Christy, you want to try? I'll guide you. It's fine.
Yeah. If so like if fluorine has like a high En, then it has a higher ionization energy because it's less likely to give up an electron because it's has 7 electrons, so it needs 1 more in its valence electron to complete the octet rule.
Perfect. So I'm going to summarize what you said. If pulling electron is hard, right, it's going to take more energy, right? It obviously implies, right? I'm going to switch it up, and this is the first time I'm going to add some jargon just to make it a little bit more difficult, get us thinking. It implies that your work function, right, or another way of saying that is how hard you hold the electron, right? So really all you were trying to get at was these 2 very simple ideas, right? You told me a lot of words, kind of like an MCAT passage. But I had to sit there and I had to listen and say, oh, what you're really trying to tell me is electronegativity is really about the pulling of an electron, right? And the real 2 ideas that are really important to remember when I think about electronegativity or ionization is it's going to take more energy if it's harder to pull the electron off, which implies that it's got to be holding on to, like, implies how hard you hold on to the electron is high, right? That's kind of what you were saying. Is that the gist of what you were trying to get at, Christy, or did I miss anything?
Yeah, that was what I was trying to say.
All right, cool, perfect. So now does everybody kind of understand that? Because what we were trying to do is we're trying to understand, remember, an informational passage. And I haven't told you all what an informational passage is, but I have hinted that it's about scientific information that you should already know, right? So we're basically doing a really quick under 7-minute chem-phys practice passage that we're making up right now. And the idea is if you're following along and want to kind of understand the exercise we're doing, we're going to walk together through how an MCAT test maker might actually make an MCAT chem-phys passage, right? And so together we've decided this passage is going to be about electronegativity. If anyone's lost, just say something in the chat. All right, I see nothing, so we're going to move on to the next slide, right? So this is kind of how I break down. This is my list, and I'm going to show the list so that you can read it as I talk through kind of the basic structure of a chem-phys passage, right? So remember, we're going to make up a chem-phys passage by the time we're done about electronegativity, and specifically about ionization energy, right? So let me transition. Let me transition slightly. Let me just kind of start talking more the direct lecture way you kind of expected. I wanted the first part to be a little interactive, get you guys a little in a different space than you were expecting to be, and then we can talk about the stuff that you came to expect to see, right? My strategy, if you want to call it that, right? So all I know is that chem-phys, the most common passage type I'm going to see is an informational passage, right? What's the whole goal behind an informational passage? It's in the name. They plan to inform me, or really even better, they're trying to check if I'm informed, right? What that means is they're really trying to give me straightforward presentation of scientific information. So they're going to give me the information. What does that mean? 1, I should already know it. So I already know what the most electronegative molecule is, fluorine. I already understand the trend lines. I also understand how electronegativity ties into energy of ionization, right? I get that if I'm going to have something that's got a high electronegativity, it's going to take a lot of work to pull that off. You guys already get that. So what the MCAT is going to do is they're going to provide you with an informational passage That's looking to check if you're informed about this straightforward knowledge, right? And what they're gonna do is in the passage, they're first gonna provide you with the main topic, right? So I'm gonna provide us with the main topic, right? I'm gonna say the main topic is finding the mass of a protein, right? I just decided this is the main topic because for some of you guys who've been on another session with me, you'll hear that I give out what I call major keys, which to me is just a time for me to say something to alert you that you should probably stop and write down some notes, right? So what I'm going through is I'm talking about the first thing I do when I get to a passage, right? So first I know what kind of passage is it. It's an informational passage. You're gonna inform me about something I should already know. It's gonna be presented in a fairly straightforward way, right? I just have to figure out how they're doing it. I just have to crack the code if you wanna use the pun. So step one, I gotta figure out what the main topic is, right? Step two, which I think is the most important part, right? I gotta ask myself, what's the specific part of the main topic they're really looking to dig into, right? What's the part that they're specifically looking to analyze? Or maybe what are they looking to determine? Right? Or what is their hypothesis? Right? These are all words that I think are major keys that are very common in passages, and we'll try to find them, that tell us the specific part of a main topic, right? At the moment, you guys don't quite understand, at least it hasn't been clear, I haven't given it to you clearly why finding the size of a protein is the specific part of a main topic that has a main idea that is electronegativity and ionization energy. Once somebody figures out how to connect those two, just unmute yourself and just share your aha moment. If you already have it, share it now. If not, we'll walk through this process, okay? So I like to start things from the top, come all the way back down. So I'm gonna clear out my notes. We'll go back from the top and just kind of make sure we have an understanding of where we are. We're trying to understand what CP passages look like. I told you that they're information because they're gonna inform you about something. It's gonna be pretty straightforward actually, and most importantly, you should already know the information from your content review, right? So the passage isn't really intended to check whether or not you know your content. It's really whether or not you can critically reason through the passage. Obviously, a lot of you guys get that, you're here. So how do you tackle this? First, you figure out what's the main topic talking about, right? When I say main topic, I like to think about main topic in two different ways. I think of main topic as the process or steps that are being described by the experiment, or the concept broadly, that's being discussed. Does anybody want to kind of jump in here? Maybe Jaleesa. Jaleesa, why don't you jump in here and kind of help me explain what I mean when I say a main topic can either be a concept or it can be like a set of steps or a process described in an experiment.
So yeah, so I guess if you had a passage talking about, like, for instance, like the cell cycle, that could be a main a concept, but then maybe the step or the process could be mitosis or how it's replicating.
Ooh, okay. Did you read the passage that was passage number 25 that I had up?
Yeah, I did.
Okay, good. I can tell you did, right? So that's wonderful, right? So I'm going to summarize what she said, right? A main topic could be either a concept, right? So the simple idea of cell cycle. It can be fact-based. And a quick trick that maybe you might have noticed, maybe you haven't. Anytime the MCAT makes the main topic a concept, they usually actually give you everything you need in the passage. right? So if they're going to give you a passage, for example, about G-coupled protein receptor and cyclic AMP, they might actually give you the picture of the whole pathway right there, which should tell you that the real goal of them testing you is to see what your critical reasoning skills are. On the other side, there's the processor steps, right? So for example, it might still be about the cell cycle, right? But this passage now might actually really be about how do you study the different steps? How do you know when the cell is in a specific step, right? Or how do you know when a specific drug that's a cell cycle inhibitor, where in the cell cycle it actually inhibits, right? So now you can understand, in order for me to answer any of those questions I just mentioned, I need to go and analyze something. I'd have to determine something. I might create a hypothesis. For any of you guys that have done any microbiology lab work, I might create an assay, right? I'm taking time because these are major keys that are literally words that you should highlight every time to again tell you not what the main topic is, cell cycle, but the specific part of the main topic that's going to be consequential to whatever passage you're reading. All right, so I'm going to stop here. And Victoria, you want to go ahead and hop on and try and kind of give us a summary of what we've done so far. We haven't gone far. We've really done three things, right? We said, what's the MCAT really trying to get across when they give us a Chem Phys passage, so described it. And then we talked about kind of like main idea, what's the main topic and the specific part. So just kind of hop on and tell me what your summary or like how you would go tell your best friend after you left this session what I said about those things.
Yes, so the informational passages, they're either about a process or a concept. And for the concept, they usually give you all the information you need in the passage.
Wonderful, wonderful. So I'm going to just point out one thing, right? The real key you missed is here and here, right? You have to understand the purpose of the passage, not what's in it or what you have to go find, right? Your plan started at step 2 or maybe 3. You didn't stop and say, if this was a patient, right? This is you like jumping into the patient's room and you're starting to talk with them and not even looking at their name, right? You got to start at the beginning. So I say that because if you understand the moment when I was studying, I understood like, oh, Chem Phys, it's an informational passage because they're literally just going to give me information. It's not going to be new information. It's really going to be information I know, right? When I mean information I know, it's just a concept, right? So they're going to give you the idea of electronegativity and the idea of ionization energy, right? The challenge is you have to take these kind of steps to critically reason through the experiment. But the way you kind of unlock, and this is a big word I've been using this week, prioritize. What are you going to try to prioritize? Your studying, your schedule, and your review of content, right? The number one thing that you can do, and Victoria, thank you very much for kind of joining me in this conversation, is understand every aspect of this exam, right? Get what the CP section is really about, right? All they're going to try to do, again, I'm going to keep repeating this because I'm trying to get home a simple point that is going to be, as all things, driven home very well when we do the passage that I'm describing here. So I'm going to go through this really quickly just for time, right? So we first want to find the main topic, right? We then want to ask ourselves really like the specific part of that topic that is really important. And then really key is we want to ask ourselves, what's the outcome that's really being considered here? And then how are they going to measure it, right? It's really important, right? What's this passage about? What specific part of the background is important? What outcome are they researching and experimenting on, right? How are they going to measure that outcome that they're interested in, right? And then the last thing you're going to do, right, is you want to make a list of your independent variables, all right? What are they checking here? Are they checking how heavy the protein is? Right? So are they checking the mass of the protein? Right? Remember I told you that what I was looking to do. Anybody remember what I said a couple slides ago at the very beginning? I said I was looking to analyze something. Anybody remember what I was looking to analyze in this fake passage that we made, where the main topic is electronegativity and ionization? Oh God, I just completely lost my spot. Miss Caruso, would you mind joining along and kind of answering and helping me out right quick?
So the only thing that comes to mind is just analyze the
because I said it. So I actually said it in the, like, while I was talking. I'm trying to see if anyone caught what I said. I was looking to analyze something. So this is a fake passage that I think it was Victoria and I started making, right? Because she's the one who told me let's do electronegativity, right? And then I said we were looking to analyze proteins and their fragments, right? What I told you is what I was specifically, I got specific right there. So make sure you follow along, because you and I are going to transition to an actual practice passage and do it together, okay?
Okay.
So I told you the specific thing I'm looking to analyze. Remember that was a major key? I told you I'm looking to analyze something to do with proteins, right? And I told you this idea of electronegativity. There's a lot of things I haven't told you yet, but I want to make sure that you're good on those pieces, okay? All right, cool. So we're going to then transition to this paragraph passage. Have you done this passage before? Have you ever seen it before? Did you look at it today?
I've had it in the Q pack. Yeah, kind of recently.
Okay. So why don't you take a look and tell me what's the specific part of the main topic that they're really digging into, and tell me where I should start. Like, read it out loud when you find the specific part of this passage. Like, what's the specific thing that this experiment is focused on? And if you can't remember, just think about the major key I told you.
So I guess what I think that the main topic is proteins and analyzing their fragments.
Yeah, exactly. Perfect, spot on, right? What I was trying to get you to see is that if you just looked for the word to analyze, it tells you exactly what this passage is about, right? They're trying to figure out the size. It's called size situation because we're not really clear on exactly how they're going to do it. They're trying to figure out the size situation of proteins and like whatever fragments they make, right? So if they make fragments, we can assume that they're going to use what kind of enzyme. Anyone on the line that's kind of, let's use this to review a little content. What kind of enzyme would they use if they're looking to make protein fragments?
A protease.
Proteases, right? So the learning point here is, you said, I believe one of the learning objectives you had here was to get better at critical reasoning, right?
Correct.
So one of the critical reasoning tips and tricks is anytime you see the word to analyze, it pretty much tells you like really what specifically is being talked about, right? If you're really trying to get to the specific part of any passage, just look for that. So guess what I look for anytime I'm tutoring students and they give me a passage to work with them? What word do you think I look for to figure out what the passage is really about?
Hypothesize, analyze.
Exactly. Those words, exactly. Those words literally, if you, next time you go do a passage, start off by trying to find the words to analyze, to determine, to hypothesize, right? Because if you go here, right, even without looking at the rest of the passage, I know, okay, cool. They're really looking to see like the size-ish of these proteins, right? So we're gonna do a quick word association, everybody. What biochem topic does that remind you guys of? What I just said, like the whole size of proteins. How would you find
techniques?
What kind of techniques?
Laboratory techniques.
What laboratory technique are you thinking of specifically?
Oh, well, Sds Page.
Ooh, Sds Page, right? Tell me a little bit about how Sds Page works. So I have a gel. And then I've got this stuck to something. This is going to be a battery, right? Let's say we call this our positive end, our negative end of the battery, right? What's the ultimate variable or chemical property that an SDS page really is trying to decipher? Anyone on the line want to say?
Charge.
Charge. What else? What else besides charge? If I show up and I have a protein up here, another protein down here, right?
Size.
Besides charge, what else about them would be different?
Size.
Size. All right, so I heard size. Somebody explain to me why size would be one. All right, so
it makes them be able to move through the gel differently depending on their size.
Oh, perfect. So I'm not sure who that was, but do you want to just kind of give us a full, kind of thorough explanation of how an SDS page works? Your best guess, however, because you were right. Everything you said was spot on. I just want you to just summarize it before we move on to the next step.
Yeah, I guess the larger the size, the less they're able to move.
The larger the size, the less they're able to move. All right. So can somebody want to take on the role of scribing some quick facts? Because I keep having to erase. Who wants to volunteer? Just shout out and say you're going to keep track of maybe 2 facts only.
I don't mind.
All right, thank you. So I believe what you said is when size goes up, they can't go as far, right? So I think that was Jaleesa, write that up. And now what about charge? Whoever wants to answer, right? In this experiment, if charge goes up, what happens to how far these things can go? If the thing gets more positive, is it going to want to move up anymore? So more positive, right? If something is more positive, is it want to stand near the negative end?
More close to the negative end because opposite.
Okay, so that basically tells you that when you're asked a question about the charge part of an SDS page, you have to stop and ask yourself, okay, is it the thing that I'm being asked about? Does it have a glutamate or a lysine? Is it negative or is it positive? Because the direction it's going to move on the SDS page depends on whether or not it's attracted to the positive lead or the negative lead. Okay? So I want to stop here, and honestly, if anyone has any questions about SDS page, the very basics of what we talked about here, please let me know right now, because what we're going to do is we're going to hop onto the passage and I'm going to read it, and we're going to kind of walk through how I process a passage like this to kind of see if we can sort of try to show you by me doing kind of the main lessons I'm trying to get across. So any lessons about SDS page? If not, can I continue? Let me get a yes in the group chat, 2, and I'll continue.
Well, the only thing I have a question about is SDS page codes it in a negative charge.
Right. So this was more of a teaching moment to get you to understand this concept, but yes, you're right.
Right. So that's why I was confused when you were asking like about if it's more positive charge, and I'm like, but
perfect. So who's this on the line? Ms. Curso. Okay, so I'm gonna answer your question in a very simple way, right? Okay. While speaking, this is what you were doing. You were listening like this, right? Unfortunately, you need to be listening like this, right? These are percentages of your allocation of time, energy, whatever you want to call it, right? And the reason I specifically say this, right? Ms. Caruso, do you mind if I call you Ms. Caruso? I don't want to butcher your first name again.
I suppose that's fine.
All right. How many passages are normally on a MCAT practice question? Like this one has 6, right? That's pretty much all of them that you usually have, right?
Right, 5 to 7.
5 to 7. Let's call 6, right? Are you familiar with the term pseudo-discrete?
Yes.
All right. How many pseudo-discretes do you usually see in a practice question?
Not that many.
Yeah. Can we say about 2 maybe usually?
Sure.
All right. Anybody else want to come in and pipe in on any other number besides 2? I believe the average number of pseudo-discrete questions that you see on an AAMC practice passage is 2, but I'm willing to take any other numbers. All right. 2 questions, right? So these pseudo-discretes are all about content, right? Only content. you kind of need to know the information to get them right. You agree, Ms. Caruso? Sorry. Why don't you try to get me to say the name one more time so it's easier? It's
Irena, and
Irena.
The last 25 years, how I tell people how to remember my name is you point to your ear. Irena.
Ear? Oh, Irena.
There you go.
Irena. Okay, so cool. So Irena, the point I'm trying to make is only 2 out of those 6 questions are going to be discrete. Right. The other 4 really require critical reasoning and are an implicit, and the other one is an explicit, right? So not hard math, that comes to like 67%. It's critical reasoning. It makes sense because this is an exam that doesn't really care about content. It's really, can you clinically reason? If not, don't show up to the first day of medical school, essentially, is what this exam is all about, right? Yes, there's a lot of other parts of your application, and I do not think that being able to perform well on MCAT is indicative of whether or not you should be or can perform in medical school, because you grow and evolve as a professional learner, but the skills that you need to actually achieve on it are very reflective of the clinical reasoning that you need to do. So this is a very long way, Irena, I actually pointed to my ear, of answering your question about the Sds page by saying the point of the Sds page is the following, right? Now let's go to the passage and I'll actually drill down why I even went to Sds page. As I'm reading this passage, right, I'm gonna read them and I want you to pay attention to what I say, how I say, and what I don't say, because this is my internal monologue said out loud as I read this passage. All right, some real big word, ionization. I know that word. Soft ionization technique. Okay, cool. I know that is like electron knock off. I kind of know the word fluorine because that gives me like general trends. All right, I keep reading and then say, okay, the in conjunction. Okay, cool. I pause because that's telling me like these 2 things are together. So let me pause and understand how these 2 things relate. So it looks like the first step, they pull an electron in step 1. And in step 2, they're kind of like checking the mass? Question mark? Not quite sure. But, oh, wait, major key. So I'm going to make this in a different color, blue. They're looking to analyze the protein fragments and peptides. Okay, cool. So at this point, I can pause and I think about it, but I've done enough experiments, enough passages to remember that, there's that one weird passage type that I commonly see all the time. I've seen this before, sometimes in chemistry, sometimes in even in biochemistry where they have some kind of light that's going to do some kind of ionization, right? I've seen this passage before, right? What's that ionization going to do? It's going to make an electron, right? A free one. And then I always see that they tend to do like this whole E thing here. And because of that, let me just make sure I remember there's like the force equation, F is equal to QE. I just got to keep that in my mind in case they ask me a question about it. All right, cool. Let me continue reading. So they're going to coat something. All right, so coated. All right, cool. So they tell me that the plate is coated. All right, cool. So it sounds like what they're telling me is that down here is whatever this DHB thing is. Great. I look at the DHB thing, nothing rings. All right, I keep reading. The coated plate is subsequently. I like the word subsequently because it gives me chronology, which is sometimes a hard thing to pick up on critical reason science passages. This came first, this came second. If I know subsequently, I kind of know what part of the cause and effect equation I'm in, second half of it, right? So if I pause and I go back to the top and I read this again, I say, it looks like they're talking about ionization, or well, nope. They're talking about an ionization technique, right? And what they're really talking about in the ionization technique in that first sentence is this piece of what I drew, right? So this looks like this must be MALDI. Kamanthi, how did you know that? I just went back to the passage and I remembered they told me something about ionization here. I knew that the DHB was coated here. Oh, so this must be what is getting ionized. That's why they coated it on the plate. Why did they coat it on the plate? Oh yeah, because they need some light to ionize it, right? So they needed some ionizing radiation. Really, all I'm trying to do is I'm trying to piece this thing together for you in real time. Ionizing radiation gets me to remember, oh yeah, I remember energy of a photon equals HF. I'm just gonna write that down or keep that in my head in case they ask me about it. Okay, cool. So far I'm gathering that they're basically trying to use some kind of light, right? So if I'm thinking about my potential variables, I don't know if these are all the variables that they can give me, but if I look at this, right, they can give me the type of light, right? They can give me the sample of whatever material is on that plate that's getting ionized. They can give me the electric field that's being used, or because I remembered Sds Page, I remember, maybe they're trying to use this to kind of figure out the mass charge ratio of a protein, which sort of lines up with what I saw that they were looking to analyze. So maybe now maybe I found a direction of where this passage is really going. All right, cool. I'm gonna read ahead. Oh, wow, look ahead. That kind of looks just like what we were talking about. So we're moving in the right direction. So we then come to the second paragraph, right? I always recommend, even for figures and that are experimental setups, read the description, because sometimes the description like this, right, doesn't tell you anything, but sometimes it does, all right? So at this point, I've gotten to the last paragraph and I read carefully. So reading carefully might be the mistake that some of you guys are honestly not doing, because the AAMC has decided each word where the comma is in order to test your real critical reasoning. Can you read a sentence and break it down? All right, so we begin the last paragraph. So this biological molecule, M, I'm not sure what it is. And one mistake that some of y'all do is y'all stop and y'all ruminate or think about what biological M is. I don't. I'm like, all right, I don't know what that means. All right, it got adducted. I don't really know what that word means. By free proton. That I know. Why? Because I remember this passage at some way had to talk about ionization energy. And if you recall, at some point, we'd even mention that ionization energy and electronegativity are kind of related. So now I've read these first three words and I don't know what protein M is. I can guess maybe protein M might be the sample I mentioned that they might be looking at, right? So looking at one protein versus the other, because the passage told me that they were looking to analyze something about protein. So maybe they have protein A, B, C, D, so on until they got to M, right? So the last part is, let's just kind of take it home. It looks like these free protons adducted, and I'm not quite sure I understand what that word adducted means, but I look here and it almost looks like they're trying to tell me like a proton came off of whatever, the DHB, I think, right? That's the stuff that was on the plate. And then I see a plus, so maybe that means that they were added together. I'm not quite sure, but let me just take a look at the experimental setup to just kind of see what's going on. Okay, let me see. 1, interesting, 2. Oh, cool, that kind of lines up as if they're trying to tell me that the DHB, which is this stuff here, was ionized here by some type of electromagnetic radiation here. And then after it got ionized, it got stuck onto these here proteins, right? It's like stuck on the proteins. So I think to myself, maybe they're trying to go protein amino acids. And I say, okay, cool. If it was a proton that was lost that had to attach to an amino acid, so maybe let me just remember glutamate and aspartic acid in case I get a pseudo discrete. Okay, cool. I'm gonna continue and say, okay, let's read the next sentence. The ions leave the MALDI plate. Okay, they're moving. All right, cool, they're leaving, moving. I don't know what that means, but I remember physics. Maybe something to do with acceleration, maybe something to do with force, not quite sure yet. But I do remember I've seen the experimental setup before, and I already put in my head that equation in case I need to do it, right? And right now, all I'm thinking of now that I've gotten here, because I see something that looks like electricity, is, so I've got electricity and I've got charge. This is really the best equation I think of, right? So let me kind of think about it for a second. So, okay, if the charge is bigger, those things are probably going to end up going faster, or the force is going to be bigger, right? If the field is bigger, you could also do the same thing. So I kind of got an idea of the relationship that is being talked about here. Then I read forward, and I see, okay. They tell me how far it traveled. I pause. Does the force, have I seen the word distance anywhere in any equation? No, no so far. I'm not quite sure how to relate distance to anything, but I'm going to highlight it anyway. They tell me that it's a uniform electric field. I kind of assume that from the picture because I haven't really learned anything that's not uniform for the MCAT. And then they leave me with this last sentence, or these last two sentences. They tell me that the velocity of the ions is inversely proportional to the mass charge ratio. Okay, so that tells me if velocity goes faster, if it's getting faster, right? The ratio of mass to charge, this whole thing must have gotten smaller. I'll say to myself, okay, cool. It's a fraction. I'm not really sure what that means. Let me break it down. For that fraction to get smaller, what's going to happen to the top? The mass has got to get smaller. And what's got to happen to the charge? The charge got to go up. All right, I got an understanding of that sentence. And I've got an understanding of how velocity relates to mass and charge. Mass and charge. You remember we talked about this idea of Sds Page at the very beginning. Because what we've been really talking about this whole entire time as I've been reading this is simply the same passage that we came up with at the very beginning. A simple concept that should already be informed about. And with that, I'm going to just stop right here and take questions about any aspect of this passage, any aspect of my approach, anything at all, because I hope to spend the next 15 minutes, which will be hopefully our last 15 minutes, walking through a very creative learning experience in order to hopefully teach you a few things about the cell cycle and hopefully to drive home some of the lessons that I'm trying to get across to you today about critical reasoning and about you prioritizing your time on really making sure you understand kind of the logic behind experiments, the logic behind content, and the way the two merge. So I will pause myself here for, you guys have been semi-lively, semi-not. So I'm gonna just give myself 2 minutes to fill y'all out. If y'all are not gonna say anything, I'll continue. Or I can call people out. We can do that. Angie, why don't you give me your initial feedback? Just about a minute or two of just initial thoughts.
Honestly, I feel like my brain is a little, like it's everywhere at once.
I did that, right? Tell me what are the big things that you picked up? How did I talk about the experiment? What words did I use? Did I use jargon?
No, you definitely focus on words that we should have already learned in content review. So you focus on those, and then
let me pause real quick and jump real quick. Let's dig in. How do you think that helped me?
Because it helped you focus what the main topic or idea of the passage was going to be.
Yeah, I mean, it did a little bit, but the main idea really of why I didn't really focus on the jargon, right, is I wanted to avoid myself getting confused by the passage or overwhelmed, right? If I say receptor, what are some things that kind of flash in your head?
Receptor, so cell membrane.
What else? Just rattle them. Let's go. Free association real quick.
Okay, so ligand, binding, second messenger.
Okay, where are receptors usually found?
Cell membrane.
Cell membrane.
Nucleus.
Is it fair to say things bind to them too? Is it fair to say sometimes they get activated? And is it fair to say sometimes they signal downstream to something?
Yes.
All right, let me ask you if the term GCPR43 minus means anything to you.
It means that it doesn't have the GPCR receptor.
Right. But do you know what that means to me? It means one word, or two words really.
Without.
No receptor. Right? And the reason I'm trying to make this, the point I'm really trying to make is a very subtle point that is about how you think about, I can't write, your working memory. For some of you, you've had I call it MCAT fog, where you take a passage and you almost don't know what you read. You forget it by the time you get down to the very bottom. You just feel like somebody put a mask on you and just spun you around, right? Is that,
Yeah.
if anyone's ever, you've felt that, right?
Yeah.
All right, what's the average number of, like psych-social review, average number of working memory items that you can keep and still be good?
It's 7 plus or minus 2.
Plus or minus 2, right? What happens if you overwhelm your working memory?
I feel like you won't be able to remember things as much.
You won't be able to remember things as much, right?
Mm-hmm.
Have you ever had situations on a MCAT practice passage where you just couldn't remember things as much on
All the
what you read on the
time.
passage all the time, right? So what
Yeah.
mistake do you think you're doing right now? Like a simple, very unconscious mistake you're probably doing?
Trying to take in everything.
Everything, right? So let's just go ahead, me and you, we're gonna read this passage, okay?
Okay.
But I only want you to read the things that I highlighted as if they were sentences, and read them out loud if you're comfortable.
So just the highlighted portion?
Just the yellow highlighted portions of
Okay.
it.
So gut microbiota implicated in the regulation of host metabolism, metabolism of dietary fiber by gut microbiota, production of short chain fatty acids, important energy sources for the host, act as signaling molecules, bind and activate G protein coupled receptors, GPCR expressed in white adipose tissue, generated GPCR43
Oh,
deficient.
stop. What did we say when we say GCPR deficient? What is that actually in your head?
No receptor.
Right, so read that again.
So generated no receptor to analyze the contribution.
Well, so just to make it makes more sense, just researchers generated no receptor mice.
Okay.
So that it like makes a little sense.
Researchers generated no receptor mice to analyze the contribution of receptor to energy regulation.
All right, so pause. This passage, what do you think the specific point that they're trying to get at in this passage right now?
The receptor.
Right, why don't you read me the sentence statement where they really tell you what they're looking to specifically dig into.
To analyze the contribution of GPCR43 to energy regulation.
Perfect, awesome. So let's take a step back and I'm just gonna draw out and tell me if I'm wrong on what you read so far. There was some gut, they broke some fibers down. Did that sound familiar? They did that somewhere?
Mm-hmm.
Some catabolism went on?
Yeah.
And then what did they produce when they broke them down?
Short chain fatty acids.
Okay, and then what those short chain fatty acids are important for what?
Activating GPCR.
All right, activating GCPR, but we're gonna remember, yeah, we don't really need to use GCPR, so we're just gonna say receptor on in our head. So receptor on, and where is this receptor found?
White adipose tissue.
Right, so fat cell. Right, and so let's predict if the receptor is on or off. Right, fat or skinny is the response that I want on either side. We're just gonna make a prediction.
So if the receptor is on, it's skinny. Then
Or if it's off,
it's fat.
fat. All right, cool. So pick up reading when you left off and just read starting with the wild type and the GCPR43, but attempt to just say that using words besides those two words.
Okay, so receptor and no receptor, standard diet or high-fat diet, body weight in grams was measured. Mm-hmm. Researchers examined the relationship between receptor function and insulin sensitivity.
Sorry, pause. Let's just stop right here because these are two experiments. What would you say the outcome they're looking to measure? And a major key to tell you where to find your dependent variable is where it says to measure. So what's the dependent variable that they're looking to measure? You obviously it's not right behind it. It could be in front of the word to measure, but
it's body weight in grams.
Am I back?
Yes.
Okay, awesome. Sorry about that. Okay, where were we at before I left off?
The last thing I heard was the dependent variable.
Okay, so why don't you first catch me up to what we've done so far, because I kind of forgot. So walk me from the beginning of the passage and just kind of catch me up to where we were at so far.
So we talked about that having gut microbiota leads to the catabolism of fiber, which leads to short chain fatty acids, which activate the receptor, and then that leads to fat breakdown. And then if you have the receptor on, you're skinny. If it's off, you're fat. And then
Awesome.
we figured out that the dependent variable was body weight.
Body weight. How did you figure it out? You tell me how you figured it out, and I'll tell you how I figured it out. Let's see if we figured it out the same way. How we found out the dependent variable.
So I just looked at the y-axis of figure 1.
All right, awesome. So that's one great major key, right? If you're ever stuck on a practice question, right? So do you want to just pass along the baton to somebody else, and we're going to run the same exercise you and I did, but from a different angle?
Yeah.
All right, somebody else. First of all, before you hop off, what did you feel about that reading of the passage?
It was a lot easier just focusing on the highlighted parts.
Only on the highlighted parts, right? So real quick before we get you off, right? This was the main idea. You didn't find the specific part until you got to here, right? All this technically is the main idea. And this is what I meant by a process or steps can actually be the main idea. You found the specific part when you found the to analyze part, right? You started to figure out one of the independent variables up here, but we actually asked you to find the dependent variable first, which is right here. How did you find it? It has to be where they're about to talk about the measuring technique, right?
Mm-hmm.
And then the real nice trick is I like legends, because legends like to tell you what the independent variables are. So you can find that one independent variable obviously has to be either standard diet or high fat diet, which to me just simply tells me diet has to be one of the variables. All right.
Mm-hmm.
And then if I continue with that, right, what is wild type versus GCPR? I guess I told you the answer. What independent variable is being tested there?
So having the receptor and not having the receptor.
Receptor or not. Perfect. So one way to simply distill all this first passage is, it's really saying it's about receptor. Well, really it's about metabolism in a fat cell. And what's specific part about it? The receptor piece of this whole story. Right? And what they're going to do, they're going to check to see if it is on or off, right? But they also mentioned that they're going to do a little side experiment here when they're going to try and do diet as the experiment. And for diet, they're just going to look and see, right? Is it high fat diet, lots of fatty acids, or fatty acids, or if it's just standard diet. Does that summary help kind of synthesize together sort of the 2 steps to this process, right? The part where you sort of read through the annotated version, which made it easier for you to focus, kind of free up your working memory. And the last part where we just kind of bring it together. Because next we're going to do, the next person that hops on, we're going to do some data analysis, okay?
Okay.
All right, awesome. Thank you so much. Any other thoughts you want to share? The reason I asked for this is everyone that's on the call is here because they're searching for something. And if you have an insight today, you might be able to pay it forward by sharing, oh, wow, I learned X. I used to do this. Now I think I'm going to do Y. That could help somebody else. So last opportunity to pay it forward if there's anything that you can kind of think of that you have taken away so far from you and I kind of talking back and forth about this passage.
So I think drawing out the diagram of the steps will help to keep everything in order. And then also, also looking for the words like to analyze, to measure, to hypothesize, to kind of focus on what the passage is really talking about.
Okay, awesome. Thank you. That's actually really great, right? Those are great things to do, right? I personally follow in your camp. I like to draw out my outlines either mentally or on paper. I like to go for my little buzzwords. As you can see, I've got them squared off, right?
Yeah.
And then I'll leave you with one little tiny advice, right? Do you highlight when you read?
Yes, but I highlight almost everything, so it doesn't really
Right.
help.
Perfect. So I will advise you when you highlight, as much as possible, begin your highlights or end your highlights with a verb. It'll make life a lot easier. You notice how many of my highlight phrases either began or ended? Should I choose another color to make that a little bit more clear?
No, yeah, I understand.
Right? So that's a high yield highlighting tip for all of you guys here. Just get in the habit of kind of, if you take a look, examined, incubated, uptake, measured. Yeah, that's about it. Right? Literally every single highlight that I have here has that. And from a very practical perspective, right? You want to read MCAT passages with the cadence, especially in biochem, that has a bit of this thing did this thing, right? That means it's a sentence that ends in a verb. Or this thing was done to you or to this protein, right? It's a simple technique, but high yield technique to keep you from getting overwhelmed. Okay, so thank you very much. Anybody else want to step up? And if you're going to step up, I'm going to ask you to kindly bring your white coat with you if you're going to step up. Meaning, get ready to play doctor. Like literally, we're going to play doctor. And kind of go through an exercise that is partially this MCAT passage, partially cell cycle, partially a med school conversation about hematology oncology. So anybody interested in either pediatrics or anybody interested in hematology or oncology, step up and let's chat about this passage together. It's going to be the last thing we're due. And really the focus, the learning objective of this passage is to really get home across the main point of this presentation today. This exam you're taking is about your ability to clinically reason. And so what I'm really asking is for a volunteer that's prepared to not only help me in this passage, but for me to guide you through an exercise of clinical reasoning.
I can volunteer.
All right, so we got two people. Let's get both of y'all on. Who? I think I got Victoria and who else?
It was Jaleesa.
Jaleesa and Victoria. All right, both of you guys ready?
Yeah.
All right, so we're gonna split up this job, right? So we've kind of looked here, and has anybody picked up on the fact that this passage kind of is talking about cell cycle?
Yes.
Right? You see words that remind you of cell cycle on the screen, in the chart?
Yeah, definitely in the chart under the group. Yeah, metaphase.
Where is that? Kind of walk me through. I can't find it. Help me find these things, right? So this is part of the exercise, right? I saw mitosis.
Well, you said in the
Right.
chart, so I went down to the chart.
No, I'm saying all over the passage. We're basically the three of us. We're gonna find words that make us realize that this is a passage about cell cycle. We've got a couple we saw in the chart, right? I'm gonna highlight another one, microfilaments, right? Because I remembered they're part of like the apart phase. Apart phase or something. I don't actually remember all of it, but you guys are going to help me remember the cell cycle because it's been a while since I studied it, right? So part of the exercise today is between the two of you, we're going to actually review the whole cell cycle, right? So learning point of this exercise is you want to get comfortable by doing more passages, right? But when you do them, you got to be looking for patterns that help you when you look at a passage to really look at it and be like, oh, okay, I see what y'all trying to do here. Y'all, like eukaryotic cells, thinking about cells. I see the word mitosis, cell division. I see microfilaments. I remember something about that. I look at the chart. It kind of confirms that this is a passage about the cell cycle, okay? So are we all good on that? Like what this passage is kind of really about?
Yes.
Anybody unsure? Because this is an important thing. We need to be on the same page. We don't really know what the passage is about. We just want high level because we're going to actually switch to a whiteboard and talk about cell cycle. Okay, how comfortable is everybody with cell cycle, the both of you?
I'm pretty comfortable with it.
Okay, cool. So we're gonna. Where do you mind if we start with G1?
Yeah, go ahead.
All right, in G1, what's the cell's primary priority? Or let's stop here. Let's stop here actually first. Victoria and Jaleesa, in order for this exercise to work well, I need you guys to take a second and kind of picture yourself in your white coat. And I want you to share with us what kind of aspiring doctor you are, because we're gonna actually work through the passage and everything as if you're wearing your white coat and you're a little student doctor, okay? It sounds corny, but trust me. All right, so think about putting yourself in that mind frame of your clinician now, and just for fun of it, tell us what kind of doctor. I want to be a gastroenterologist or a GI focused surgeon, so maybe bariatric surgery. So that's a little bit about me. Real quick, come on guys, just real quick. Let's just kind of get this part out of the way.
I guess I want to be some sort of pediatrician.
Pediatrician, so you like kids. That's wonderful. Yeah. Okay, awesome. And who else?
I want to do primary care.
Primary care, awesome, cool. So where you got a pediatrician and a primary care doctor and a guy that wants to do GI, maybe surgery, right? So we're gonna start this whole passage somewhere else because just like energy level wasn't feeling it. So we're gonna go and we're gonna actually be doing casework, right? We're in our didactic lesson, all right, guys? And we have to make up a case. So the way cases are structured, there's an age, right? And there's a demographic, right? So we heard pediatricians. So somebody give me a number between 8 and 15.
10.
10. Give me a demographic, just general description, as whatever that means to you.
Female.
Female. We'll add other description, right? So 10-year-old female, and let's get a name.
Jan.
Let's get a name. Huh? What's the patient's name?
Jan.
Tan. Okay, cool. So I'm getting ready. I'm sorry?
I said Jan.
Jen or Jen?
Jan.
Jan. Okay, cool. So I'm going to present the case of Jen. Jen is a 10-year-old female who's been on the ward, the pediatric ward, because she's 10 years old, for the last 2 weeks. And she's here because she's got acute lymphoblastic leukemia, guys. It's not good. And our job, I believe Victoria and Jaleesa, right?
Mm-hmm.
We've got to find drugs for her. We got to pick drugs for her. That's what we have to do during this morning's meeting. What drugs are we going to put on her for chemo? Okay. So Victoria, tell me a little bit about cancer. What do you know about cancer? Basics.
I know that the cell cycle just keeps going and going, like there's no regulation.
Cell cycle keeps going, right? So keeps going. So you and I are going to talk about cell cycle then, okay? Since our patient, what's our patient's name?
Jan.
How old is she?
10.
And what kind of disease she have? She has leukemia, right? Some kind of blood leukemia, right? You kind of sad for her?
Yeah.
All right, you're sad for her too, all right? So let's start with G1. What's the cell's main goal in G1?
Growth.
Growth, right? And really it's just preparing to do what?
To duplicate the chromosomes.
Perfect, divide, right? So the next thing is S, and S obviously is what? Divide, right? And then there's G2, right? What are some priorities in G2 you think?
To prepare for mitosis.
Right, so kind of define that to me in a more simpler, less jargony way.
Keep growing.
Keep growing, right? Because you need to get more and double everything so that you have enough when you go through anaphase, right? Mm-hmm. And that's about it, right? So we've got the first three things, right? And if I told you, which maybe you do, maybe you don't, there's 3 checkpoints or 2 checkpoints here. There's checkpoint 1 here. Checkpoint 1. I know that says chapter 1, but you get the point. If you happen to know the name of the enzyme or the protein that is at that checkpoint, it would be a lovely extra kudos to you. But if you can't think of that, just help think through what sort of goal the cell would have if it wanted to stop and check this whole process between growth and division of its DNA.
Just make sure that everything is going properly, like there isn't a problem with any of the organelles or the chromosomes or anything.
Perfect. So you said organelles, chromosomes, check, right? So this checkpoint is called, have you ever heard the term P53?
53?
Yeah, has that word ever bounced in your head before? P53,
No.
anybody?
No.
Anybody else on the call heard anything about P53? Want to just hop on and help us out?
It's a tumor suppressor gene.
Ooh, I heard a good term, tumor suppressor. So we'll save that for a little later, okay? But tell me what the whole point is of this P53, right? I'll tell you actually for the sake of time. Its job is to sit there, check if DNA is broken. If so, pause and fix. Kill if can't. Right? This is literally what an all P53 thinks. Why? Victoria, what did you tell me the priority was in G1? You're preparing to do what? Divide, right?
Mm-hmm.
So part of preparing to divide is you want to make sure you check and see if anything got nicked in your DNA. You know, maybe, we don't know, Jan might be living in a household where her mom is a smoker. And all that smoke has been damaging her DNA. And we got to make sure that we check to see we don't have any mutations before we divide. That makes sense?
Yes.
All right, cool, awesome. So that's enough of this back end stuff. Now let's go into mitosis. I will kind of start here. Jaleesa, you want to help me out with mitosis?
Sure.
All right, cool. Mitosis, first step of mitosis is what?
Prophase.
Prophase. And what is the priority for the cell in prophase?
Prior to the cell, basically the nucleolus disappears and...
There you go. I like to think about it as prophase is just really like the big thing is nuclear disappearance, right? There's a lot of facts you guys know about prophase, right? But I'm telling you the facts that I still remember after a year in med school. This something to do with the disappearance of the cell member, of like the nuclears, right? Why? Do you think that they make a lot of drugs for this step, if I don't remember it?
No.
So they don't make a lot of drugs. Do you think there's a lot of clinical research on prophase step in cell cycle?
No, it's mostly probably about metaphase or anaphase.
Boom. So do you think the MCAT is really going to make a exam practice question about prophase as a passage with an experiment, or are they going to put it where they can actually find research papers about it? It's a fact. The passage that we're going to see is about prophase, metaphase, and anaphase, and telophase. And Victoria, in case I've lost you, what was the first thing that we started off with, right? We're in our didactic, right? We got our white coats on, right?
Yeah.
And so we're trying to figure out how we're going to treat a 10-year-old female who came to us with leukemia. We got to pick drugs. He told me she's got cancer. So he said, hey, cancer means the cell cycle keeps going. So what are we going to, what's the goal of our exercise here? What's the main idea here that I'm talking about? If we were talking about in passages, what am I really trying to get at if I want to pick a drug? Anybody can answer this, to stop cancer. Logically, not science, just logic. Anybody. Jaleesa, you want to try?
Yeah, you just have to pick the drug that's going to effectively stop one of those like steps in the cycle.
One of those steps in the cycle, right? So Victoria, what do you think about that as kind of how we're going to approach our didactic to try and treat our patient? Try to pick a step and try to stop the cell there.
Yeah, we'd probably find a drug that does something with anaphase or metaphase.
Okay, cool, perfect. We can even do some of these other ones. We're not going to talk about some of these other ones right now until the very end, but let's first ask this, right? If I was going to circle, do you remember seeing the word microfilament in that passage that we did earlier? The actual one that we looked at, number 25, the term microfilament. Can you relate, Victoria, you sounded pretty confident in the cell cycle, right? Relate microfilament to the cell cycle for us and kind of explain to us what role microfilaments play in any aspect of the cell cycle we've described.
Isn't it like to split the cell in half?
To split the cell. Okay, cool. So if it's going to be used to split the cell, what action are they doing? Use a verb. What verb word do microfilaments do
Divide?
in the cell? They not quite divide, right? So think about the picture in your head that you have of the cell cycle, right? What are those microfilament fingers doing in metaphase?
Pulling apart.
They're holding them at the middle plate.
Oh, metaphase, yeah.
In anaphase, what are those fingers doing?
Pulling the chromosomes apart.
Pulling the chromosomes apart. So these blue cells represent where microfilaments are involved. Jaleesa, do you understand what I mean? What we meant by these are the two spots that microfilaments are involved?
Yes.
What do we mean?
Well, so in the in the cell, right before so anaphase, there's a plate in the center, and there's the filaments that are pulling it apart. So that's where it comes into place in the cell.
Perfect. And so basically they hold it and then they break it apart, right? Cool. So I'm going to add in a new piece of information. And are you guys familiar with the terms polymerize, and depolymerize? Anybody want to explain those two terms really quickly? It shouldn't be too hard. My spelling is terrible, guys. I'm sorry. Anybody want to just explain? Victoria and Jaleesa, you guys have been answering a lot of questions. Anyone else want to try and ask? Christy, you've been a little quiet. Haven't heard from you in a while. Hop up and let's talk about polymerization and depolymerization.
Yeah, is polymerization combining?
Coming together. Okay, and what's the other one?
Depol is the opposite, so it's breaking off, coming off.
Okay, perfect. All right, so then let's try to connect these two, right, to what's going on over here. Whoa. What's going on to the right side, right? So the reason I said this is that microfilaments, right? This is outside information, so don't memorize it, don't write this down. This is what the passage is gonna give you, are little balls, and when they polymerize, the balls stick together and they make a rope almost. When it's time for you to take them apart, they depolymerize, okay? So now I want you to think real quick, Christy, metaphase or anaphase, which one is going to be the depolymerized side and which one is going to be the polymerized side?
Would the depolymerized be anaphase?
Why?
Because in anaphase, they're breaking apart.
Breaking apart, and then what's going to happen right after they break apart?
They go to like the opposite side.
There you go. So do you need them anymore after anaphase? Not until the next cycle, right?
Yeah.
Exactly. So there we go. We just needed to do a little quick review, get some critical thinking going on. So thank you very much. So Jaleesa and Victoria, you guys ready to come back?
Sure. Yeah.
Yeah.
Right, cool. So let's bring this all together, guys. So remember, what was our patient's? Does somebody want to present our patient actually?
I can.
All right, go ahead.
This is Jan. She's 10 years old. She's presenting with leukemia.
And what's the task at hand? What are we trying to do now?
Stop the cancer.
Stop the cancer. What's the kind of strategy we're going to use to stop the cancer?
I'm going to find a drug that targets part of the cell cycle.
Cell cycle. And what two steps of the cell cycle have we decided on?
Metaphase.
Perfect. So on the right side, I'm just going to visualize something to kind of help y'all make sure y'all get this. That if you gave it a drug that stopped it at metaphase, it will look kind of like this, right? Because they're stuck in the middle, right? If you gave it a drug that had to do with anaphase, right? Anaphase, what's happening in anaphase? They're getting pulled apart, right? So you might end up with a situation where they're like, oh, almost got pulled apart, but one of them got pulled apart, one of them didn't. Who knows, right? Either way, that cell is not happy. Neither is this cell, because they're both stuck. Are we fair on that part?
Yes.
Okay, cool. So we've actually figured out what drugs we're going to give her. We're going to give her a drug called vinblastine, or we're going to give her a drug called vincristine. Vincristine sometimes goes by an older name of cytocolazin. And these are drugs that are big drugs for treating hematology, oncology. Why? This stuff is irrelevant. This is the part that you need to pay attention to. They block actin or microfilament function. And Jaleesa, if we stop microfilament function, what are we going to stop the cell from trying to do? what steps in the cell cycle are we going to stop?
We're going to stop at metaphase.
Metaphase. All right, cool. Thank you very much. All right. So ladies and gentlemen, we have come up with a whole plan, and our patient is actually going to recover. Fun fact, leukemia for children these days, about 80% recovery rate. So we don't have to worry about it. Everything's all good. All right. So now let's try and relate it back to the passage. All right. So before we go in, what did you guys feel about wearing that white coat? That white coat felt good? Thinking through stuff?
Yeah.
Yeah.
Felt good, right? So Syed, or let's go Victoria, because Victoria's been here long enough. I don't know if you were caught following up on everything. So Victoria, you up for kind of reading the passage that we have again, and then try to just read it quietly. Actually, everybody, we read this passage carefully, right? And then whenever you guys are ready, I want us to start a discussion amongst all of us about the passage itself. And I don't want us to necessarily talk about the passage. I want to talk about the clinical reasoning exercise we had right before coming back to this passage. So let's take about 5 minutes on the clock, actual 5 minutes, and then I'll just check in with you guys. Either the way I want us to run this is either I'll check in with you guys at 5 o'clock, or if you have something to say that's on your mind, unmute yourself and let's begin to have a 5 minute conversation, 5 to 10 minute conversation, because this is really the end of my lesson today. Right? This passage, having you understand why I gave you the exercise that we did with white coats, how it relates to this passage, and what you can do with that information to really kind of hopefully go do well on your MCAT. So this is kind of where the lesson ends, and this is where whatever learning you have left, whatever questions you have, you can ask them. So I'm going to be quiet until the 6:45 mark, and we'll go from there. Okay, quiet group. So I'll just go down the line. Angie, what are your thoughts about the exercise we did and the practice passage that you see in front of you?
What do you mean? What about the exercise?
So what did you take from the exercise? What were we trying to do? What did we start off trying to do initially?
So you work with what you were presented with and then try to work backwards to figure out.
Well, let's even talk specifically, right? We had a patient that had a disease, right?
Mm-hmm.
And what did we have to do? We had to pick a drug that did what to the cell cycle?
That stopped it, prevented it from
Stop
dividing.
it, right? Are you watching on the screen?
Mm-hmm.
We had to pick a drug, either vinblastine or vincristine, that would stop the drug either in metaphase or in anaphase where cytokinesis occurs, right? So you might imagine at some point, some pharmaceutical company ran this experiment. Let me stop the drug, let me stop the cell at these 2 points. Let me introduce rather these 2 drugs, right? Because remember, what do these 2 drugs do? They affect the function of what? Microfilaments, and to a certain degree, intermediate filaments. Why are those important? Because some of the function that these microfilaments are involved in is obviously cell structure, so forth, but most importantly, the separation of sister chromatids during mitosis. Why is that important? Because in our exercise, we had to pick a drug for our child. That drug had to stop the cell cycle, and we selected metaphase and anaphase as our drug targets. We understood these would make good sense drug targets because if we stop the cell at this point, if we froze it there, we could basically kill the cancer, right? So now you've got a target. Now you have to find a vehicle to deliver that payload of drug target. So the way they did it in this passage, they said, let's attack the microfilaments. Why? Because I know the microfilaments have to do 2 things. They can either polymerize or they can depolymerize. Polymerize. Right? And I believe it was, who is it that told us that the depolymerized side was closer to anaphase? The polymerase side was closer to the metaphase side, because that's when the filaments have to come together to do their job, polymerize. Anaphase, after they're finished, you don't need them anymore, they need to go away, polymerize, metaphase, right? Because it might make sense to check out whether or not two drugs, two independent variables, vinblastine or vincristine, how is it that they affect if you look at group, what this means is where the cell's actually at. All right, so that is literally the end of this passage, the end of my session today. I wanted you to do a simple thing. One, put on a white coat. Two, perform some clinical reasoning skills, not critical, clinical reasoning, in order to understand the cell cycle a little bit better, but most importantly, to be prepared for the MCAT, which after today, between this exercise and between everything I've said repetitively, you should recognize is a challenge that requires you to somehow prioritize 67% of your time to critical reasoning development and about 33% of your time to content reasoning or content skills. All right? If you can keep those really 4 things in mind and add one last thing in order to round it out to 7, that when you read passages, make sure that you actually think about how are you going to worry about not putting too much information in your head, not overwhelm yourself, not get MCAT fog, avoid overwhelming your working memory. That is it. And if I can leave you with a parting word, prioritize if you're studying for the MCAT, especially if you're in the cycle. Because if you focus on prioritizing, you'll actually be able to find time to really, really put 67% of your time into what's important. So with that, thank you very much for hanging out with me for about an hour and 45 minutes. I always appreciate giving out these free sessions to kind of give people a chance to kind of see and understand a different perspective to MCAT studying. And that is all I'm going to do and talk to you. So at this point, if you have any questions, please message them to me, not message them to me. I just talk out loud unless it's something private. If so, I'll put my email at the end. If anyone can see this, right? Does this look like a picture of the cell cycle, right? Victoria and my 2 white coats. This looks like kind of what we talked about, right?
Yeah.
Guess what source I got this from? Do you think I got it from my MCAT prep or from my board prep work?
Board.
Look at all these drugs that y'all got to know when y'all get to med school.
Wow.
What's that one right there?
Vinblastine.
Where's that from? Where did you first recognize that from? You recognize that on the MCAT passage, right?
Yeah.
I hope my point got across by me showing that, right? That was a TPR passage that's testing a concept that is core at your understanding of oncology on your boards, i.e., can you understand the cell cycle? Can you understand the central dogma? Can you then go through the critical reasoning steps to get that if you're gonna knock out cancer, you gotta knock out you gotta either knock out the replication or you gotta knock out the function, the cell's ability to make anything, right? Again, just kind of driving home the fact that you really, really, really need to focus on your critical reasoning because it's going to prepare you when you get to med school, okay? Are these sessions provided every day? No, not really. Right now, they're more ad hoc, unfortunately, because I started med school this week, but they're going to be coming a little bit more formalized. Unfortunately, we didn't get a chance to do any physics, and this kind of turned more into a critical reasoning session. I hope you guys got some stuff out of that, but I have an email list that I try as much as possible to sort of keep up with and email out information once a week. So on Sundays is when or Saturday night is when I typically send out an email that kind of tells people what my expected free sessions are going to be, right? What is it that I think is on my mind? So today's lesson that you guys sat in on was actually created last night as a result of me